For those not familiar with our webinar series, what we're doing is we are presenting in basically 3 blocks today. So the first block is going to be about primer design, and this is going to be part of our larger DNA assembly webinar series here. I'm just getting a laser pointer up. You can follow my laser pointer. And so my name's Dennis Mishler. I'm a professor in Austin, Texas at the University of Texas at Austin. And I'm going to be with you for the next half hour or so, something like that. So, boom. Primers, DNA primers, really important. And so I'm going to talk to you about why we use them, what they are, what factors to consider when you're designing your primers, and a little bit about how you can go about designing them online on apps and how to order them. All right. So hopefully last week you guys saw the basic molecular biology introduction, and we talked a little bit about PCR there. And that's what we use primers primarily for. We use them for PCR. And there's a lot of different types of PCR reactions you can conduct. You have this figure here from Thermo Fisher, and you can see they talk about all sorts of different applications for PCR and different types of PCR. And so they all use primers, and sometimes they use different forms of primers with different natures in terms of their sequence. And so the primers you design are going to vary greatly depending upon the type of PCR you want to conduct and your template DNA. There are always exceptions. You can use primers for things that aren't PCR. You can use templates that are maybe not DNA. But we're going to talk about, generally, we're talking about using DNA primers for PCR. Okay. So what are DNA primers? Well, the DNA primers are short oligonucleotides. And oligonucleotides, if you're not familiar with the word, simply means many nucleotides. Oligo, many nucleotides. And DNA primers are going to be complementary to the DNA templates that they're interacting with, right, that they bind to. Our example over here, you can see there's a DNA primer, this red FP Ford primer here. It's got this red box. And you'll notice that it's bound to the template DNA. And it has this red little half arrow here indicating its directionality, right? So it's going in this direction into the sequence. Now remember, even though it's shown on the top here like this, the primer is not actually base pairing here, right? The primer is actually base pairing to this to the reverse strand on the bottom here. So this G with the C with the G, right? So this is just a basic introduction of what DNA primers are. We're using them for PCR. They should base pair to a complementary strand. You need 2 primers, right, for a single PCR reaction, and the primers need to be pointing towards each other, going this way. All right. So when creating DNA primers, you have to consider a number of factors. The first one, the important thing, is what is the purpose of your PCR reaction? If you're making, if you're just doing a sequencing reaction, or if you're amplifying DNA from a genome, or you're doing some various diagnostics, you may just want to make fully complementary primers. That means primers, like on the last example here, that completely base pair to your template sequence. And those are common, those are just common primers. They get used all the time. Those are fairly easy to design, and we'll go through that in a little bit. However, there's also these partially complementary primers, and these are used a bunch when we're doing mutagenesis or cloning strategies in other various diagnostics. So if you have a DNA sequence from a genome somewhere and you want to alter it or mutate it, you could do that through DNA synthesis, where you order a very large gene straight from a company, or you could potentially do that through PCR, where you add the mutations while you're doing the PCR reaction. Okay. In the example over here on the right, let me just move my mouse a little bit. There we go. Over here on the right, you can see we have 2 primers. And interestingly, there's 2 big differences relative to the previous example. The first difference is not all of the primer is in that red box, right? If you go back here, you can see everything was in the red box. Everything was part of the primer that base paired to the template. Here, these portions of the primer are not base pairing. They're not complementary, right? Those are some overhangs on the primer. And that tells me right now we're going to be doing some mutagenesis. We're doing something different with this sequence. You can also see there's a red A here. That's a mismatch. It's a mutation. It tells me that we're also introducing mutation with this primer. So we're going to be mutating this T position to an A. Okay. Same thing over here. We're making a mutation here. So this is a partially complementary primer. We're going to be doing some mutagenesis, and we're going to be adding some restriction sites so that we can do cloning with it. The other thing I want to point out about the primer is notice the arrows over here and over here. So these primers are actually going opposite directions. So this tells me these are not meant for a single PCR reaction. This primer here, the reverse one, is going to be going this way, and we'll need another primer over here to make a PCR product, while the other primer, this forward one, is going to be going past the screen, and we'll need another reverse primer way over past the screen. And so what's happening here is this is an example where we're going to be taking a known DNA sequence and creating 2 PCR products with mutations and then stitching them back together. So we're going to be doing some cloning. Right. So I want to talk about some of these features though that we have to take into account with regards to the actual properties of the primer. All right. So we have to take into account the length of the primer and the GC content. So there's just properties of the primer. We have to take into account the melting temperature of the primer. So that's an estimated temperature for what the base pairing between the template and primer will be. So melting temperature is the temperature at which half of the primers will not be bound, essentially. Okay. And the annealing temperature is related to the melting temperature. During the PCR reaction, we need a temperature where the primer will fully bind to the template. And so that's based upon the melting temperature, right? So your melting temperature, if it's 60 degrees, your annealing temperature is going to be lower. Generally, I like to say 5 degrees lower. So if the melting temperature is 60 degrees, our annealing temperature in the reaction, we probably want to start with around 55 degrees just to make sure the reaction is going to work. We have to consider the secondary structure of the primer, and I'll show this in a little bit, and I'll talk about that then. And there could be a lot of other factors depending on your purpose for the reaction, okay? And I'm not going to get into those details, but depending on the enzyme you're using, depending on other factors, you may need to change some other detail about your primer, okay? So where to design and order primers. This is actually a really important thing. A lot of teams don't realize they can get these primers or DNA sequences in general free. So there are a lot of options. I recommend going to this link here. This is the iGEM partners page. And you're going to see there are a lot of different companies that will provide you with stuff, including free DNA. I've just highlighted here IDT, which is one of the bigger sponsors. You can see on the right-hand side here, it tells you what their deal is, and you can click there. You basically can get about $2,000, $3,000 worth of free DNA. That's what this comes down to. You can just get free DNA sequences, primers, full genes, whatever you like, okay? Go to the partners page. There's also stuff from Twist and some other companies as well. For the rest of this webinar, I'm going to be using Benchline and IDT to demonstrate how to make primers. All right, and we're going to make 1 or 2 examples depending on how our time goes. Okay. So let's make a primer. I'm going to go ahead and post in the chat these sequences here. If I can get the chat back up, I'm going to post those links. And these links here are links to shared sequences on Benchline. And you guys can look at these now or later. You're not going to be able to edit these sequences directly. These sequences are not editable when they're shared, but you can copy and paste those into your own DNA file if you want and edit and work with that. Okay. So like I said, we're going to go ahead and do this now. We're going to do this demo. I'm going to have to change my screen share to the correct one. I think it's this guy. Okay, and hopefully this transitions over. Great. So this is the IDT website. We're going to use this in a little bit. This is the Benchline web app. It's a cloud-based system. This is my preference, but you can use whatever you like, of course. And move some stuff over. There you go. And so what we're going to do here is I'm going to use this DNA sequence to make some primers. All right. And I don't want to go over all the details of Benchline. That's just something you're going to have to learn or explore on your own. But once you have Benchline up and running, you have a DNA sequence. Here's the name of the sequence. You can see it's a red fluorescent protein for iGEM demo. So that's something I made here specifically. And we're going to make some primers. We're going to make 2 primers. We're going to pretend we're trying to clone this whole gene. So if we have this full-length sequence here, we're going to make a forward primer at this end and a reverse primer over here, which I already have made. But we could make a primer anywhere. If I wanted to make a primer here, I could do that. It just depends on what our purpose is. All right, so I'm going to go ahead and start by making a primer over here. I'm going to make this forward primer. Make sure I get that correctly highlighted. Great. And when I highlight this, I'm going to just point out a couple things. First off, you can see the start and end of the highlight, so 1 and 23, the total length of the selection, 23, and you can see the GC content. And that's really important, right? GC base pairs are stronger, and so they will contribute more to a melting temperature. So the more GC content you have, the higher your melting temperature will go. So if you're having problems with a low melting temperature, try to find some GC base pairs, get that up. And then lastly, we have this melting temperature part here. This is for the selection. So if I change the selection, you'll see a lot of the numbers at the bottom there change. So the end will change, the length, GC content, melting temperature, all these are going to start changing, right? And that's just related to the length I have, okay? But I'm going to go with 23. I chose this because I often like primers around 20 nucleotides long, and I like to have melting temperatures that are around 60 degrees or a little bit higher, all right? Now, if we click on melting temperature, because you may see a different number than I do. If you're working on Benchline right now, you're not going to see that number possibly. And the reason why is you can change the melting temperature based upon how much magnesium you have. So here, if I have 0 magnesium, which is sort of a default 0, you'll see my melting temperature is a lot lower, 55.7. But I know in our reactions, we usually have at least 1.5 millimolar magnesium in our reactions. And so I'm going to just change that setting, and you'll see this melting temperature number is going to pop back up here. Boom. Okay. So this is a good start. So we have a sense as to what our melting temperature is. That's within the range I like. It's at least 60, and higher is fine normally. And the length is fine in my opinion. And so now I need to make the primer. I'm going to right click here. I'm going to create primer. I'm going to go in the forward orientation, right? I want it to go into the sequence. Click. And now I've opened a new tab. I'm in this design primer tab. If I click back to sequence map, you'll see I have this now, this primer being formed. It's dashed. That means it's in the process of being made. It's not actually made yet. If I go back here, I have the sequence of my bases. I'm gonna have to save it somewhere, so I'll figure that out in a second. I have the 3' location, so that selection for the primer, it tells me where it's positioned. I have an overhang. This is gonna tell me if any bases, I'm gonna tell the system, these bases should not be base pairing. So I'm gonna go ahead and put that at 5. And I'm gonna say the first 5 bases should not be base pairing. When I do this, if I go back here, you'll see now that the box ends right there. So these nucleotides are not base pairing anymore, and that'll just be sort of indicated there. But let's go back here. Let's go ahead and change this back to 0. And now we'll see the box goes back to the whole sequence, right? Boom. Okay, so that can be very useful. If you want to add in like an enzyme for a cut site or something, that would be part of your overhang if you're trying to make some changes. All right, so we have everything in here. Now, everything here looks pretty good. We got to give it a name. I'm just going to call it Demo 1 for fun. It's going to be saved here. That's perfect. We're going to check the secondary structure somewhere else. I'm not a big fan of the Benchline secondary structure check. I'm sure some people love it. I really like the IDT one, and I'm going to go there in just a second. So this looks good to me. I'm going to go ahead and save the primer. Okay, and now you can see it's fully dashed, right? It's a full box. It's a full real primer. It's there. If I click on the primer itself, it'll highlight exactly that, give me that information. So I now need to copy the sequence. I'm going to go to IDT. I'm going to copy the DNA sequence in this orientation. And I'm going to go to IDT. And IDT is a site where you could order the primers from, but it also has this really cool tool called the Oligo Analyzer. And this tool can be used to help you find things like melting temperature if you want, or secondary structure, or other considerations with the primer. And I'm not going to get into all of those because of time. So here I have my sequence. I pasted that in. Notice my magnesium concentration. You could change this. I'm going to put this back to 1.5. And I'm just going to go ahead and hit Analyze. And it's going to tell me some properties. Notice it has a melting temperature. Here's the hairpin. Okay, is the hairpin. Okay. And in the hairpin here, what we can see is a secondary structure. So if we come down here, you'll see this information. And if we click on this image, this is a prediction for how the primer might base pair with itself, how it might interact with itself. And if the primer interacts with itself too much, it can't interact with the template, right? And so there'll be a competition between primer binding to itself versus primer binding to the template. And so here, if you see circles as are base pairs, that indicates more structure within the primer. And if you have a lot of that, that's bad. 2 is not a big deal. 3 is not a big deal. So none of these sequences here are really of a concern. But you can see the different structures. Just click on it. The other thing it does, though, that's really useful is it gives you an estimated melting temperature for that structure. And so you can compare this temperature here, this TM here, which is about 30, you can compare that to your own TM for your primer. And if those are similar, that's a problem. But if they're really different, you're good to go. And my general rule of thumb is 10 degrees Celsius. So if this melting temperature for the structure is 10 degrees less or more than that, so in this case, if we're going with, if I find the sequence again, sorry, 63, if that melting temperature for that stem loop for that primer was like 53 or lower, I would say it's not a big deal. I'm not worried about it. And in this case, I'm not, all right, because it's like 30, right? So 30 is a lot lower. That structure should not be present or interfering with the reaction. Okay. Okay, so that was a real quick run through of making one. I want to show you guys real quick this second sequence here. This is a reverse primer. It's got a couple differences here. First off, I've added some overlap sequence or overhang sequence here, and this sequence is not base pairing to the template. And I want to just highlight this in case you're trying to work on this ever. If I delete these A's here from the sequence, these are just extra A's to show you something. Notice the primer disappears. It's not gone. It's just the way the system works is it doesn't show it. But if I add these A's back, primer's back, okay? So that overhang sequence is nice because it doesn't interfere with your calculations when you're trying to figure out melting temperatures, okay? Now, if we want to take this primer sequence and look at it on IDT, we actually need to do it with the primer tab here. We want to get the right orientation, which is starting with just right here. So we got to do it this way, copy this, hopefully that copied, and we can post up here. Now, here, we want to just do the hairpin again, right? We can use melting temperature on Benchline, but hairpin. And notice what happens in this hairpin, structure here. The melting temperatures are a lot higher, 50, 53, that's pretty high, right? And you can see it's because there's more base pairing occurring, all right? And this is potentially problematic, right? That might cause problems. Now, my general rule of thumb, again, is 10 degrees. So if we go back, we can see this is 53 here. And we can see here, the melting temperature is estimated to be about 63. So it's probably okay, this primer is probably okay, but if I did the PCR reaction and I did not get product after a couple of attempts, I'd be very worried this primer was not good. One of the easiest solutions for that, though, is just to make the primer longer on the inner side. So if you just add a few more nucleotides here, you'll pump up the melting temperature, and often that'll be enough to get around the problem. Okay, you can just increase that a little bit if you need to. Okay. Okay. Now, guys, again, this is just a general rule of thumb. This is not always going to be the case in every single set of PCRs or every set of primers. So do take that into account. All right. So again, example, we just went through that there. I showed you a couple sequences. I'm not going to talk about this one here. I'll talk about this PCR product in the next block of the webinar because we didn't have enough time. And so from this, hopefully you learned a little bit about the important factors in terms of making primers, designing primers. And please utilize your free resources, okay? IDT, Twist, any of the other companies, they've got stuff there for you, okay? It's okay to order whole G blocks or whole genes. That's fine. You don't always have to have primers. Primers are a good option. We're going to get to our next webinar in a few minutes, but we're going to pause right now to take some questions, and then we'll get into Golden Gate assembly, which is a really awesome technique, okay? So I'm going to go ahead and exit out of this. And I will stop my screen share for right now, and we'll see if we have any questions from folks.
You can write in the Q&A or even speak up or raise your hand at the side, and then you can we can allow you to speak up. Looks like everything was clear, Dennis.
Uh-oh, that's a bad sign. If you guys have questions, though, feel free to ask here. We're happy to answer them, even if they seem simple. If you don't have questions, you can always email us later. I have that email up on the webinar, and this will be up there as well. We're happy to answer questions or go into things. Have you guys I have a question for you guys. Have you guys designed primers before? You guys can tell us in the chat or wherever. No, okay, yeah. Well, if you guys start designing primers and you need any assistance, let us know. We're happy to help you guys out. You guys can come to our office hours, which are every other Tuesday, or you can always email us at that email address, okay? All right. So, oh, here we go. Is there a new question? Okay. So could you please tell us why magnesium concentration is same 1.5? I use, so that's a concentration I use because all of our reactions will, in my group, all of our reactions will have at least 1.5 millimolar magnesium. That's something you can figure out when you look at your PCR buffers. You can look at the PCR reaction buffers you use, and in there it'll tell you the concentrations. And based upon that, I know there's always at least 1.5 millimolar magnesium. Yeah, so so I got a question about the 10-degree difference. Why does that happen? So let me get into that one first. So the idea here is your primer has to base pair to your template. If your primer folds up on itself instead, if it starts interacting here, it can't interact with the template. So any structures that are seen within the primer itself have to be fully melted when you're doing the annealing temperature. Okay. So if your melting temperature is, let's say, 65 degrees Celsius, our annealing temperature is going to be 5 degrees less. That's just a general PCR thing in terms of our procedure. So if our melting temperature is 65, our reaction's running at 60, our primers are going to be bound to the template. That's great. And if we have a secondary structure within the primer, that secondary structure needs to be fully melted when we do our PCR reaction. If that's not fully melted, it's going to cause a problem. Okay. Yeah. All right. So for your overhang on the reverse primer overlapping, the A's, did you use a specific overhang sequence or does it not particularly matter? Good question. So on the template there, let me share that screen. I like that one. I like that one because I didn't talk about it and I had thought about making sure. So here, this sequence here is not actually part of the template. I made that up randomly just to show the overhang. In terms of the primer, right here, these 3 A's on the primer, that can be pretty much anything. It's randomized to protect the restriction enzyme site here. There's a restriction enzyme site here, and to allow the restriction enzyme to work better, you usually want to have a few extra nucleotides on the end of the DNA sequence. So that's why I have those A's there. Some people have preferred sequences for this here, but often it doesn't matter too much what it is. Can PCR primers be used to remove restriction sites instead of adding them? Absolutely, yeah. So I actually, you can do a point mutation. So you can do mutagenic PCR, where you change a couple nucleotides within the sequence. And then you do PCR, that removes the restriction site. And then afterwards, you would have to reassemble. Here's what I'll tell you. It might be easier to just reorder the gene using a G block or some sort of gene synthesis from a company. But you can use the primers to make mutations like that. I think.
If you need any help with
I'm sorry, Renee, what was that?
If any one of you needs help to design such primers, which for example delete restriction sites, and you really want to do that, we can actually also help you in the office hours, specifically for your project, for your primers.
Yeah, and that's great. When we have, there's one last question, then we're going to move on to the next thing. When we have a higher melting temperature of our primer, you told that we can increase the length. Yes. So yes, exactly. That's a good point. By Raj. I'm not sure how to say your name. I'm gonna go with Raj. I'm sorry, I don't have the rest of it. That's right. You have to recheck the secondary structure on IDT. So when you add a few extra nucleotides, you got to take that whole sequence and put it back through the secondary structure prediction to see if it helps you or not. It just depends on the base pairing. So if you add additional nucleotides that don't contribute to the primer interacting with itself, you're good to go. If those bases, though, do interact with the primer itself, it's a problem.